Okay, this morning we're going to continue the discussion on conversion factors, length, weight, and maturity. Hopefully Jed will be online so we can ask him some questions. I don't see him there yet. However, we can start the discussion now. Does anyone want to start off with a question or a comment? Yeah, Dan.
Yeah, hello. Just a quick one. I mean, yesterday it seems, with Simon's presentation talk a little bit about spatial variation growth, but there seems to be evidence in the spatial variation in the length weight relationship as well. I mean, IATTC, we recently did quite a bit of work on the albacore tuna. Kind of people have looked at some of the data samples collected. There's kind of strong evidence show the the variations length weight relationship on regions. So there's question raised of how to deal with that, put those things in the stock assessment. But I mean, when we had those discussions, just feel it's quite hard to do those kind of thing in the stock assessment, right? Unless you're doing multi-stock models. In the single stock models, generally quite impossible to incorporate those kind of things. Probably the same reason we can't deal with spatial variation in growth in those kind of single stock models. So yeah, just don't know if any people have any experience in dealing with these kind of problems.
Yeah, does anyone want to respond to Dan's question? I mean, one thing that you can think about is the historical VPA approaches, right? Where you have the weighted age for each, well, for the combined fishery, but often you would have weighted age for each fishery. So some approaches, maybe like SAM or ASAP, maybe they actually input a weighted age matrix for each fishery. And so that would be one way of dealing with that. So you'd end up having, presumably you've got lots of age data and everything too, but you would actually come up with a spatial weighting of catch times age times length times weighted age, or whatever it is, length of age, and deal with it that way. But then you also need to have a population one, which is more difficult, but I guess that would be the same as what we're doing with the indices of abundance, where you're weighting it by the CPUE instead of by the catch. So maybe not possible in all our analyses because we don't have a lot of age data, but maybe in some. Yeah. So, yeah, Simon.
Yeah, I guess one of the biggest issues is when you've got both weight and length data in your stock assessment and you're trying to fit to both of them, and they're in different times. If you've got a mismatch, you're going to get conflict between the information and the two data sources. So in that case, you're probably best converting weights to lengths before, rather than putting them both in separately. If you can convert with the correct conversion factors, because the assessment only has one internal conversion factor. I mean, currently most assessments only have one internal conversion factor. So yeah, you need to get them both in the same currency so you don't get a mismatch. And that's, you can do that for current data if you've got good sampling information. But there's also a lot of past data where you don't have any sampling information and you never will. But hopefully there will be like seasonal and spatial patterns that are conserved through time, and then you can estimate what those are. Like say the warm pool of the CPO might have a particular conversion factor, which is different from what's up in the far north. and then you could use that. You know, use a stratum specific conversion factor to get everything into the same same currency, I guess.
Okay. So the concept there is there might be a lot of things that you might have to do outside the model to deal with spatial structure. So, Carolina, so we had some issues in our assessments in terms of conversion factors and using the wrong conversion factor, or someone else converting it before we getting it, and it didn't match up, right? So can you sort of just summarize that?
Yes, so when we did the work with Sato-san in, I guess, a couple of years ago, we started to investigate the interesting patterns we saw in length composition, so sort of shifts, blocks of shifts of data. So we scrutinized that data very well, and there were two things that happened. One, we had mix of data from different fleets, so training vessels and commercial vessels, and they had different length composition. But also, for some historical data, there was the transformation of the weight data that they collected into length, and that transformation we didn't know about, and that was causing some of the problems. And also we used for our own weight data that we had, the weight to weight transformation that was not appropriated for the data from the Japanese fleet, because the way the high temperature vessels, low temperature vessels got the fish is different than the other gilling got weight. So we had to use the WCPFC transformation. We didn't have some for us. So that's still in there. We still don't have a transformation that is specifically for our fishery. But yeah, so we encountered a lot of problems and found a lot of patterns, and some were resolved after we dealt with what we could do at that time.
Okay, thanks, Carolina. Is there someone online has a comment? Oh, Jed's there. Okay, so yeah, Jed's online now, so if anyone has any questions for him about his presentation, then we can ask it now. Yeah.
Hi everyone. Yeah, sorry, a few minutes late there, but I'm here.
Yeah, well, thanks for making it, Jed.
No worries.
Enjoyed your presentation yesterday.
Great, Mark. That's good to hear. Yeah.
Yep. Okay. So has anyone else had any experience with using conversion factors and having misfits in their model because of the conversion factor was old or borrowed from another area or anything like that? Yeah, Carolina.
Another problem is, like with the conversion factors, is that sometimes you get this distribution that are kind of like a chainsaw. So you will then need to aggregate in a wider classes because of the transformation 1-to-1. I don't know, will produce these spaces between the beans, right? So masking whatever signal you had.
But how do you normally get around that, Carolina?
So for the weight data we used, we just increased the class, the size class, so we kind of include more data in each size class. So the weight class, we increased that.
Yeah, thanks, Carolina. Yes, Simon.
I guess this is kind of on a different topic, but in calculating conversion factors, there are, we tend to assume the same conversion factor throughout the entire growth of the fish. Like the standard A times weight equals A times length to the power of B. but that's not necessarily accurate. So, I mean, there are lots of, I guess, tricks you can do in analyzing the data using a spline to fit that instead of having the that standard relationship, just to get a better picture of what's actually happening in the data, and things like when you analyze your conversion factor, not just throwing all your data in, but taking into account the structure of the sampling, like the fact that if you've got a lot of fish from the same set, they're probably quite similar to each other, so you shouldn't use all those data. Which I guess is an argument for having a more, I guess, a more broad scale sampling program to get length weight relationship data. If it's going to be important in the stock assessment, it needs to be a kind of a systematic sampling approach.
Yeah, thanks, Simon. I have noticed that often when you fit a length weight relationship, it misses at the large sizes because of the abundance of fish in the medium sizes. And it may be the large sizes that might be actually make a difference depending on what you're using that for, particularly if you're fitting to weight length data maybe.
Yeah, and there are differences between sexes as well. Like when they mature, there's a kind of a change for females, which is a bit different from males. Just those kinds of little things, and if you get most of your data at a particular size, you are going to get the extremes wrong, because as a very fixed shape, like a power curve, it's not going to, it's going to rely on where most of the data are, and be wrong towards the extremes.
Yeah, so any comments or questions?
I guess one of the things I was interested to hear about, Mark, was, yeah, just that I think I raised one question in the talk, but not formally, but it was around, yeah, trying to capture some sort of uncertainty around the mean relationship. And was that discussed yesterday at all after the talk, or were there any comments on that from the crowd?
Yeah, we actually didn't have any questions or discussion after the talk. We were going to wait for today, so you
Okay.
know, while you were here. So yeah, but there was discussion about that, I think, in the session on length frequency data.
Okay. Yeah.
Because we also fit to weight frequency, and then to use the weight frequency data, you have to use the length weight relationship. And so we don't usually take that extra uncertainty into consideration when we're fitting to the weight frequency data. So it's important to take that into consideration. I don't think anyone's taken that into consideration. It did come up at some of the reviews, though, I think. Maybe the Western Central Review, maybe. Yeah. But that will probably require some modifications of software, I'm guessing. Yeah. Okay.
Yeah, I guess at least you have to fit, if you wanted to consider at least 2 more models in a grid, that can create a lot of extra work. And I don't think it's been done at all in FCL from my knowledge. There may be others in the room who know a bit more about the history of that.
Yeah. So I guess looking at some of the focus questions here, the first one is about, is the data we have adequate enough? And then the second one is about whether it changes over time. They kind of probably mix together. If it's not adequate enough, we don't really know if it changes over time because we haven't got enough samples. But I know some of the length frequency rate relationships we're using are very old. Some of the conversion factors are very old, and maybe even the maturity. But I have a feeling the maturity ones are probably more recent than the other ones, because it just seems a bit more of an interesting topic for people to research. So we tend to collect data on that, but not on the weight length relationship. So I guess maybe we could go through the different RFMOs and see when was their last length frequency data set. But let's do that after Aaron comments.
Yeah, thanks. I was just wondering, maybe this is a question, Jed, for you or others. You know, we think about length, weight, we think about maturity. One of the things that we often take as more of a given is fecundity. And you know, there's been quite a bit of recent research. I'm not so familiar with tuna, but more broadly on the impact of atresia, you know, changes in actually the analysis through histology and things like that. And I can think of a couple of papers where, or work at least, that results in, you know, 50% or a doubling of spawning stock biomass, depending on what that, or spawning stock biomass, anyways, what that fecundity relationship is. So I was just want to throw that out there, that I feel like it's a growing recognition, at least in terms of uncertainty in our calculations. And I was curious if that also applies to tuna, if there's a growing appreciation there. So I was just thinking, Mark, maybe that would be a throw in fecundity on the discussion. Thanks.
Yeah, that's a good point to bring up. Actually, in our yellowfin assessments, we have fecundity, we have spawning frequency, is it fraction spawning maybe, and then also maturity, but that's only for that species. There's also the idea about old females' eggs aren't that good, and so the older individuals, or no, wait a minute, sorry, not that one. I've heard that one too before, but it's the one that's more popular is that older females' eggs are more likely to survive than younger ones, and so the actual relationship is even stronger. So any comments on that, Simon?
Yeah, back in sort of early 2010s, we looked at those issues for yellowfin and big eye and albacore, and looked at well, the fecundity curve having an exponent higher than 3, and that means that the larger fish generate more, have higher fecundity than would be suggested by their weight. So they're allocating more of their resources to the larger they get, the higher proportion of the resources they allocate to reproduction. And there was also, I've seen a suggestion that older fish, like their individual eggs are bigger, and so they're more likely to survive. And if you get that relationship, then the contribution of the larger older fish to future reproduction is higher, and so the value of retaining older, you know, of reducing increasing the survival of the older fish is greater than is otherwise expected, and it changes the MSY parameters. So it actually does have quite a big impact on the stock assessment and where your MSY is, where your other optimal fishing, etc. is. So the problem is that those power parameters are quite poorly known, very uncertain, and so that is a big source of uncertainty in the stock assessment.
Yeah, thanks, Simon. Related to that, Carolina did some work comparing the effect of fecundity on the reference points through the definition of spawning biomass, also versus the effect on steepness. And Carolina, do you want to summarize that? Because I can guess, but I might get it wrong.
I'm not sure if I remember very well, but I guess when we use fecundity versus weight of spawning adults, a spawning female, it was about the same. But then when we use just the proportion females, then you had an effect in both the reference points and I guess it was on this status of the stock. So just having just the proportion mature or not, it was not sort of enough. But if you had at least the weight of the females as a proxy for fecundity, then you will do fine compared to having the actual fecundity.
And also, I think the effect of the stock recruitment relationship was fairly minor compared to the effect of changing the metric of the spawning biomass definition, right?
Correct. Yes, I think that's correct.
Carolina, were you using so weight being the bigger females, more fecund?
Yes.
That was the assumption. Yeah.
Correct. Just using the weight of the female as a proxy for them being more fecund, having more eggs. Or
Okay.
Yeah. Any other comments or questions? Yeah, Carolina?
Yeah, I was wondering, like what Simon said about the larger eggs being less propense to die or survive better. I wonder if, like, with our labs, because we have the IATTC lab, if any experiment that will help us at least to build our conceptual ideas about this. And it's something we could ask them. It's not natural conditions, but maybe they can explore that to see if that is plausible.
Okay. Any other questions, comments? Yeah, Carolina?
Yeah, just following up in my own comment is, are people like looking at the size of the eggs compared to the size of the fish? That's something else we could ask the biology groups to sort of look.
Yeah.
We do have some. Yeah, we do have some access to spawning females at times in Noumea, for example, particularly on albacore. But it's nothing formal has been put in place experiment-wise. But Carolyn, I wanted to ask, what's the situation in your lab there? Do you have which species are you able to access, I guess?
Yeah, we have a captive population of yellowfin tuna in a IATTC lab in Panama.
Okay.
And it's been over, like, it has multiple generations, and there's a lot of interesting studies that came out of there.
These like relationships between size and fecundity are going to vary a lot spatially, because all of these life history parameters tend to vary spatially and seasonally. So there's a need for a lot of sampling to estimate what they are, especially given the fact that they do have a big impact on the stock status that you get out of the assessment. So I guess any biologists out there who want to get funding for their research to study this is a good it's a good motivating factor, because it can have a big impact.
Yeah, Ilario.
I think that on top of that is that, as Simon said, because they are batch spawner, even the batch fecundity and the spawning fraction can change for the individual fish along the season. So depending where you are collecting the fish, you may have a different signal of the batch fecundity that should take also into account. So I think that seasonality within the spawning season, because even individual fish that can spawn 15 times, of course, the batch fecundity at the beginning, when they have more energy, maybe can be larger than at the end, and the same at the population level for the spawning frequency. So those things complicate even more the analysis of the reproductive potential. I guess that, at least in my understanding, in most of the cases now, only maturity is being used and fecundity is incorporated in few cases in the tuna stock assessment. Thanks.
Yeah, we have a question online, Matt Vincent.
Yeah, so it seems that a lot of these tuna we've shown that they have spatial variability in growth. There's spatial variability in these length-weight relationships and these other conversion factors. And I'm just curious if that might be evidence towards showing that there might be spatial structure in the actual stocks, like different spawning populations and stuff like that. And because they are highly migratory and they mix a lot, that might not lend toward, like, because you don't need very high mixing to be in the genetics in order to be able to say that they're within the same population or whatever. But I'm curious whether we should start looking at these data more specifically and trying to see if they are different populations just based on these different relationships, and maybe look at maturity and fecundity as well.
Yeah, we do that. Well, I think Kurt Schaefer and Dan Fuller, our biologists, incorporated that information into their evaluation of stock structure, if I recall correctly. So it is something that we've looked at, but I'm not sure if many other people do that. Carolina might touch on that in the conceptual models for stock structure, so maybe we'll bring that up again then.
Matt, I'm not sure if, and everyone, I'm not sure if this has been touched on earlier in the meeting, but yeah, we're trialing a few different approaches. Paul Hamer might have more to say on this, the status of it, but using the length frequency information in sort of a classification approach to understand if there's any variation in space among different regions. So you can sort of, it's actually, I think, Haukun's package that's we're using and modifying a little bit for the big eye and yellowfin spatial structures. And we've also done some work in the past with Nicola and Tiffany Vidal on trying to cluster based on CPUE time series for longline. But there's several issues with using longline data, particularly back in the day. But yeah, I think that Matt, there is stuff going on in terms of using that information, and I agree. I think also Simon and Eric's work on just examining length-weight relationship variation across the region can also be really helpful, and maybe be tied in with things like close skin mark recapture and different genetic approaches using otoliths and other markers to build knowledge and to borrow strength from all of those different empirical data sets. But yeah, there's still some way to go, I think, to be able to have strong confidence in any one of those data sources, and maybe combining strength would be a way forward for good practice.
Okay, thanks, Jed. I think we better move on to the next discussion, but we can bring those up again maybe later in the meeting. So we're moving on to growth now, and Lisa's our keynote presenter for that. And Lisa, can you share your screen?
Yes, good morning. Can you hear me well?
Yeah, we can hear you well.
Perfect.
Good morning.
Okay, let me see.
Yeah, great. Your screen is on.
All
and
good?
It's full screen. Yeah. So
Okay, perfect.
go ahead.
Well, good morning, everybody. My name is Lisa Ayu. I'm a stock assessment scientist at NOAA Fisheries at the Southeast Fisheries Science Center in the US. So I'm sorry I couldn't be with you today. So I'm tuning in from Charleston, South Carolina, but I'm really glad to be listening to the discussions. And today I'm going to be focusing on growth modeling. And my personal background is more with ICAT, but I'm going to try to give an overview of good practices that I've picked up from across tuna RFMOs. And the list that I provide is, by no means exhaustive, but it's kind of the central questions that that stood out to me as I was reading through the different stock assessments. And some of those good practices are also, personal opinions. So feel free to disagree with me. I hope it'll start some interesting discussions. So we all know that growth is a very important part of population dynamics, but how growth actually is used inside an assessment will really depend on the assessment platform used. It can be used to translate these are flipped numbers into biomass, length-based processes to age-based processes. It can also tangentially be used to derive closely related life history parameters like natural mortality. And we saw a lot of that yesterday. And so the model type really matters when you're asking, does growth make a difference? Does growth affect our stock assessment results? In general, if we're fitting to link data, our assessments are going to be more sensitive to any errors we have in growth. And unfortunately, any error we have in growth will translate into bias in our management advice and stock status in most cases, unfortunately. So let me walk through a few examples that I've found where it really did make a difference, and then I'll go over kind of the main questions that that I want to focus on for good practices. So this first example, this is the Atlantic yellowfin tuna assessment from ICAT from 2019. And what happened is we had new information on growth. We had new validation work done on the use of annual aging data for yellowfin tuna. And we had to update the growth curve for the assessment, but that also caused a change in the maximum age assumed. From 11, which was based on tagging data, to 18, which was based on bomb radiocarbon dating of otoliths. And so because in the assessment we're using maximum age and growth to scale our mortality, it actually had quite a major impact on the assessment. And you can see on the right-hand side the different vectors, in red, the old vector, and in blue, the new one. So effectively lowering natural mortality across the range of ages. And that caused the stock status to become much rosier than it used to be. And just from that one change. So it's really important. And another similar example happened in Pacific Big Eye in 2017, where again, new age estimates came into play. And so it was time to re-estimate growth. And they found that they estimated L infinity to be lower. And so with the new growth curve and the interaction with selectivity, and the interaction also with mortality and maturity at age, because it did shift fish towards the shifted the first age of maturity towards younger fish. It resulted in a big impact on the assessment results. And below here are the Kobe plots showing the old growth curve with a high L infinity on the right-hand side. And then you see when you plug in the new growth curve, now you're in the green zone. Your stock is not overfished and overfishing is not occurring. So again, important impact. And then the last example I'm bringing up that was mentioned yesterday is in the EPO with the big eye tuna, where there's question around this recruitment shift that's being observed. Is it a recruitment shift or is it model misspecification? And so one of the sensitivity runs that were conducted was to see, okay, what if we change the way growth is being specified? And they went ahead and tried to estimate growth internally using a Richards function, and they found a lower L infinity. The model estimated a lower L infinity, and it resulted in a larger stock size, less depleted stock. You can see that on the right-hand side. And it actually reduced this perceived recruitment shift. So whether or not growth is really to blame is still, I think, a question. They had a lot of other hypotheses and interaction with selectivity, but growth did matter. You can see it makes a big difference. So the central questions I want to go over today are how flexible should the growth curve be? Second is, can annual rings in otolith be used to age yellowfin and big eye tuna? The third is, should growth be estimated internally or externally to the assessment? And then what to do about variability in growth and how to approach it. I originally had thought to talk about should ages be conditioned on length, but I'm going to run out of time if I cover that. So maybe it's something we can bring up in the discussion portion. So how flexible should our growth curve be? Well, first let's think about what data we have, typically for tuna assessments, we rely on either length frequencies or hard parts or tagging data or all of the above or a combination. But in general, our data are relatively limited, especially for tropicals compared to the temperate tunas. And they're generally restricted through space and time. And usually we have more data at the end of the time series. In terms of modeling, generally, von Brundtland-Lanthy or Richards curve is used. There is a use also of a growth cessation model in the EPO. And you see on the top right what those different curves can look like, with the von Brundtland-Lanthy being the least flexible of them all. And then you add flexibility with the Richards or growth cessation model, really primarily to capture L infinity a little bit better. On the bottom right side, you can also see that in some cases we have biphasic growth, which we talked about yesterday. And again, I think the jury is still out on what exactly we're observing. Is it the biology? Is it selectivity? Is it a combination? But we are seeing it in different data sources and different oceans. So certainly worth to keep investigating into that so we can model it properly. In general, all these growth curves are fixed in the assessment with some exploration of internal fitting during the assessment process. And then they're usually space and time invariant, and usually the sexes are combined unless they really have good data. And then the variance is generally specified as a linear function of age or length. So what are some good practices we can think about? Like any modeling approach, I think start simple is a good start. Only consider additional complexities if the data are informative and if it improves goodness fit. And the thing that we're concerned about, I think, is it has to be flexible enough to characterize the mean lengths of the most represented age groups and L infinity accurately, because that's going to influence our results. And with L infinity, I have a couple points on it, which is, we need to be very careful of extrapolations beyond the range of the data. L infinity should be observed in your data. It should be observed in your length frequencies. And if we think about it, in the sense of a normal distribution, you should have about, most of your data within 3 standard deviations of this L infinity. It shouldn't be the biggest fish you ever saw. So I think we need to be very critical when L infinity is being estimated. Do we believe it and respond appropriately? And on the other end of the spectrum for extrapolation is L min, which often we don't really think about. It's usually the L1, for example, in SS. So it's a length at a young age, and sometimes that also falls beyond the range of our data. And if you're using internally something like a Lorentzian scaling, it can actually have a huge impact on your mortality vector. And I think we forget about that, at least in my experience. And if you are fishing on those age groups, that will make a difference on the assessment results. So I think we need to start paying attention to that L1. And then finally, variability in size at age, in my opinion, should be a function of length rather than age, with growth really slowing down at old ages. I think it really makes more sense to make it a function of length, but happy to hear opposing viewpoints. Okay, the next topic I stamped controversial. I hope it won't be controversial soon. I think we're working in the right direction, but essentially what's happening in the tropical tuna world with aging yellowfin and big eye specifically is that historically we've really been relying on daily aging. And only recently have we started to develop good protocols for annual aging. And there's been some recent validation work that points to the fact that annual rings do appear to be deposited on an annual basis. And it's a combination of OTC tagging studies, strontium chloride studies in the Pacific, and BUM radiocarbon dating, which we've talked about already in this meeting. And the comparative studies between daily aging and annual aging of the same otoliths show that as fish get older, it gets increasingly hard to use daily aging, and that you see a departure between the two estimates. And daily aging tends to underestimate the the age compared to the estimated age of the annual aging. And right now, if you look across assessments, for example, in the EPO, daily aging is used, but in the Western Pacific, it's a combination of daily and annual, and in the Atlantic, it's annual aging and daily aging. So I think we can't be using different scales, and there's really a need to start doing some, or continue doing some cross-lab exchanges. I know this work has already started, so I think we're working in the right direction, but because this maximum age can be very influential, I think we really need to continue this work and you know, just agree on final aging protocols that we can all apply for the same stocks and species. So good practices that you know, annual aging appears to be the preferred aging method for large and old tuna, but that daily aging is still very useful, and it should be restricted to the smaller, younger tuna. But it also is really useful in improving precision in our age estimates. And I'll come back to that when I talk about individual variability in growth. And the use of daily aging, I think, to that purpose should be done across all tunas. Now, the next question is, should growth be estimated internally? We all know, I think, the benefits of internal estimation. You can, it allows you to integrate multiple data types quite easily. You also have consistency of assumptions between your growth model and the rest of the population dynamics and the fishery dynamics operating in your model. It's accounting for things like selectivity. It's accounting for aging error if you have an aging error matrix. You can even account for sampling design if you do use conditional age at length. And probably the most appealing part of this is also that it propagates uncertainty throughout your assessment. So why, when looking at our assessments, why are we mostly using external estimation? Well, it seems like we're resorting to that in different cases. So in the first case, when there's no data, it does happen for stocks like skipjack that are really hard to age, where there's really not much data. There's disagreement between different areas of the ocean. And so we're resorted to having to borrow from a different stock or kind of combine all the information we have and from the multiple areas or multiple stocks, and then fix it in the model. Another reason for fixing it that I've seen is model instability, where during the assessment process, the analysts try to estimate these parameters, and then they just decide we need to fix them at those values because there's too much model instability, and we need to move on with this process and provide management advice. Another thing that happens is sometimes the internal estimation is not flexible enough. For example, your modeling platform may not allow you to use a two-stanza growth, or it may not allow you to integrate tagging data, which is the case in SS right now, although it is going to change. And then the last part, which I think is probably the most important one, is sometimes internal estimation results in some unrealistic estimates of growth. And that's usually a sign of model misspecification. So of course, it's a good idea not to just accept those parameter estimates blindly, and to actually look into it and do something about it. And usually what happens is growth is fixed, and then it's kind of the problem is on the table, and it needs to be addressed in the future when there's more time to look into the issue. So what can we come up with for good practices in actually internal versus external modeling? I think it's a good idea to start by exploring whether it's possible to estimate the growth parameters inside the assessment model. If it's unstable, maybe including priors could be helpful based on either external fits or other species. And then another good practice would be very critical of the results. Are the data in the assessment informative with respect to the growth parameters? We have tools we can use. We can look at likelihood profiles. We can look at the correlations. And And then are the estimates realistic? Like I said, you know, we have the luxury with growth that we know something about growth. If I say the L-infinity is 500 for skipjack, you're gonna look at me and say, nope, it's not. So unlike steepness, we actually have a ballpark idea of what growth is doing. And if something is unrealistic and raises a red flag, then it's probably because the estimates are being biased to fit, you know, other data in your integrated assessment. And so in that case, I think it's best practice to revert to fixing the parameters to something realistic based on external knowledge, and then look into these issues of reaction, you know, reactions between different parameters in your integrated assessment. Interactions, that's the word I was looking for. And then third, I think we need to be really concerned and aware of the interaction between L-infinity and selectivity. I've looking at through the assessments, that's most often the issue, the uncertainty that causes the most difference in stock status. So we need to really think about this one, and I don't think we can think of them separately. I think we need to think about the selectivity as well. And one good practice is probably to not attempt to estimate growth and dome shape selectivity at the same time. And then I do pose a question for the group, which is, you know, do we have any reason to believe that we can't observe the largest, oldest tunas in our fishery? So if your L-infinity is higher than your length comp, and you know, maybe you have dome shape selectivity, or you're saying, you know, there's this huge cryptic biomass, you know, I could see that in the 50s and 60s, a longliner wasn't going as far as deep, you know, for example, for our tropical tunas, but is that a reasonable assumption today for modern data? Now, the next big topic is variability in growth. If we ignore variability in growth, we know that it can seriously bias our parameter estimates and management advice. And there's really four main sources of variability. There's individual variability growth, there's sex-specific variability, temporal, and then spatial. So for individual variability in growth, the question I ask is what we observed as variability in the individual growth, is that actual variability in individual growth, or is it something else that we could work harder to account for? And I think Mark mentioned that maybe yesterday or 2 days ago that, you know, for example, birth date. If we assume the wrong birth date, then we're actually causing error, and it's perceived as variability in growth. You know, there may be seasonal differences that we're now lumping into an annual time step. And the illustration I have below is work done by Jess Farley et al. So these are big eye ages, and they're generally, if you look on the left and right, they're actually generally the same samples. There's a few more on the right-hand side, but the point here is that the one on the left-hand side is a set of fractional ages that was derived with this older algorithm that was using an assumed birth date and an assumed timing of deposition of the annual rings in the growth in the otolith sections. And you can see that there's a lot of variability, right, around that growth curve. And then on the right-hand side, this is new work that they've recently done, where they're actually using a relationship between the daily increments and the size of the otolith for those really small fish to adjust and derive a more accurate birth date for each individual that they're aging. And then also taking into account more information on the edge of the otolith. But what happens, as you can see, is that you're really tightening now. You're getting rid of all this noise that is just really error in your birth data segment, and you're really tightening your data around the curve. And I give it a little gold medal because I think it's fantastic that we got rid of something that we were able to account for by just kind of being, you know, better about the way we were adjusting those ages. And as you know, working as a stock assessment analyst, we often get the data, you know, already done. Here are some fractional ages, but maybe we need to take a step back and say, how are they derived, and can we get rid of some of this uncertainty before we stick it into our assessment? Because all this noise is just going to give us a headache for no reason if we can actually make it better. Sexual dimorphism, that's one that it's funny because I was reading papers, in older papers, early 2000s, a lot of them say there is no sexual dimorphism in tunas. And then you start to look at more recent papers, and everybody starts to realize, oh, wait, there is. We just didn't have enough data to detect it. And so I don't know much about it, except that I've noticed that a lot of the times it's a difference in L infinity. So what is the hypothesis is that the females are investing more energy for reproduction, but less for growth once they're mature. And so you tend to have smaller females compared to males over those older age groups. Now, I don't think it's been said very, very much, the extent to which this really makes a difference on the assessment, whether you would consider a single sexes combined growth curve or separate growth curves. And I think we're probably also lacking the data required in most cases. But, is it something we should be starting to look at and, move towards? On the other hand, temporal variability, I think we have a pretty good idea that it is happening. And there's many reasons for it. We know that there's seasonal differences in growth. We know that there's changes in growth through time. And the three examples I have here are changes in growth for different bluefin tunas, Atlantic bluefin, southern bluefin. And mostly because they have more data. We just said, if you want to look at change through time, we need data from the past, and we don't have these data for for many of the other stocks. But we can assume that it's happening all over. There's a lowering of L infinity observed, for example, in Atlantic bluefin tuna. I think that's one that is probably very important to include. Because we just mentioned that L infinity has an impact on the stock assessment results. So a word of caution though, and looking specifically at the case where we're using OLIF data, for example, with Atlantic bluefin, if we're looking at changes in growth through time, but we're just fitting, for example, a von Brent-Lampe to different data sets for different decades, say, just the nature of the data set can affect the estimates of your von Brent-Lampe growth curve because of the correlation between K and L infinity. So if you have a lot of small fish, for example, in the 90s, but then you're sampling a bunch of, you focused on the bigger fish in the 2000s, that difference alone can cause you to see a difference in growth. So I think we need to be very careful that we're comparing apples to apples and that we are truly detecting a change in growth. And that's no criticism to that study. I was just, as I was looking at that graph, I was thinking, yeah, there's differences in the spread of the data here. And then on the right-hand side, those are mostly based on tagging data, which I think is a really good platform for investigating changes in growth through time. And now spatial variability, that's on everybody's mind. I think we can all say, yes, it's happening. It's being observed across many different stocks. Skipjack are notorious for having differences in growth between latitudes and longitudes. On the right-hand side, there's a case for albacore where they were seen to be bigger on the more easterly side of the South Pacific. And I believe Simon worked on that assessment, and that they actually did try to separate and come up with regional growth curves, and it did have an impact on the assessment. So it does appear to be important, but that's probably one of the hardest things to accommodate for in our assessments. And I have the example here at Atlantic yellowfin that I just want to point out because we talked about movement and stock structure. In the Atlantic, there was a collection of data near Ascension Islands, and there's some really, really big fish there, and they're not seen in the catch at length for other other, not even the longliners in other fisheries. So ultimately, we didn't include some of these larger fish in the assessment for the growth curve because we didn't think they were representative. And yeah, the theory is that they are kind of a resident population because we were seeing this with our tagging data. So yes, there's probably some refuges, but you know, are these fish, you know, kind of the exception to the rule? And should we look at them? Should we learn from them? And do they tell us anything about maybe L infinity back in time when there was less fishing pressure? So trying to come up with kind of good practices for how to deal with variability and growth, I tried to do this kind of logical chart, I guess, that you would walk through. And this is not final, so I just put it out here so that if you have ideas on, you know, parts that should be added or removed or moved around, that I'd love to hear it. But essentially the steps I was thinking about were, you know, start with, do we have the data to investigate the change, and be critical, you know, are these differences we're observing, are they an artifact of the differences in sampling and methodology, or are they really differences in growth? If you don't have the data, go get it. We need to expand data collection. We can't do anything without data. But if we do have the data and you know, we are seeing something, then we can start to look at it in the assessment context and say, okay, are these data informative? Again, looking at likelihood profiles, correlations, and then we can move on to say, you know, okay, we can add this complexity into our assessment, but is it justified? Is there confounding going on? And again, we know growth is tightly linked to selectivity and recruitment deviations and other life history parameters. So if there are issues in confounding, we probably need to halt and start doing some simulation work to make sure that we understand what's happening and to make sure that we can determine if it's likely to influence the stock assessment results. Now, if we're lucky enough to have this beautiful model that can accommodate complexity, and we can move on to looking at, does it impact our results, you know, through sensitivity analyses? And if it does, we have to decide, you know, what's the most likely hypothesis, and you can make it your base run, or we don't know, this is just part of the uncertainty, and then we can do some sort of an ensemble modeling approach or grid approach for uncertainty. But even if it doesn't impact the results, it's obviously very important to document it and revisit it in the future because it might eventually impact our results. So finally, I'm not going to dwell on this, but there's a multitude of ways to address these you know, we have different tools depending on our assessment platforms to address modeling variability and growth. I think the most difficult one is going to be spatial variability, and I put that little graphic here from the CAPAM growth modeling. I think it's still valid that you know, sometimes we have an easy answer if there's differences in growth and no movement, and then separate your assessments. That's a simple answer, but for the most part, we're in that gray area, and we're a little bit stuck, and I don't think we have any solution as of today. So finally, just looking ahead, I think there's a clear need to continue to expand our growth data collection and do it in a coordinated and designed fashion so that we're not stuck with violating all the assumptions that our models require. And then ultimately, I think there's we should look at similarities between stocks, but we are stuck with a lot of things being stock or species specific. What I'm really excited about is progress in aging. I think epigenetics and near infrared spectroscopy is going to allow for more extensive and representative data collection. And you know, should we kind of jump on that train with tunas, and will it allow us to do things like a large scale collection of empirical weighted age data? There's also use of tagging data to inform growth that's in development for SS. And then we should certainly continue tagging studies to parameterize spatially explicit models. And then lastly, that I think Simon mentioned that just earlier, that, you know, there's probably some environmental indices that we could be using to better account for this year-to-year variability, and is very likely shared between similar species, like similar sized yellowfin and big eye, for example, on an oceanic basin scale. So probably more work needed here. I haven't seen much into that, but it would be a really interesting way to account for it in the assessment. With that, I'd like to thank you all, and I'll take any questions if there's time.
Thanks, Lisa. You used up all your 30 minutes, but we can be flexible and ask some questions anyway. So does anyone have questions for Lisa? Carolina.
Yeah, Lisa, thank you for a great talk. Nice summary of all the issues. Sorry, I was thinking about the variability of size at age. So what you showed us is like an improvement on the estimate of the variability for when you age the fish on a day-by-day basis. And then so how do we combine those different days, let's say, for the period of, or the time step of our assessment? Because unless we, sorry, correct the length frequency to shrink them and grow them, which is what we do in the EPO, you will have to use a variability that includes the different birth dates within that quarter, plus other sources of variability. So what should we do?
Yeah, no, that's a good question. I was thinking more in the context of we get a lot of data where we're given fractional ages and then we're just converting them to a calendar age. But even that, even doing that alone reduces the observed variability around those calendar ages because the fractional age is more precise. So you're thinking like on a quarterly time step, for example?
Yes. So if we don't correct the length frequencies, we will have variability because of the aggregation we use in time.
Yes. Yeah. I don't have a strict answer for you other than I still think you would see an improvement by first adjusting your ages correctly in the first place, because then and for all these ages we have birth dates, so I don't know if there would be a way to I'm not sure, maybe make use of platoons, I'm not sure, in combination.
Clearly something we need to think about. I mean, it's very good that we have better gauging. That's very, very good. But like translating that into variability in the stock assessment might be something we might need to think about, because to our experience in the EPO, it does make a difference as well. As well as the average, the variability does make a difference.
Yeah, absolutely. Yeah.
Yeah. Has anyone else got a question? Yeah.
Thank you, Lisa, for your presentation. I have a comment for daily aging study. We've been doing captivity experiment using yellowfin and big eye tuna in Japan. And yeah, just to validate the reliability of daily aging. And it is just preliminary results, but the number of daily wings doesn't match with the days in captivity. So there could be large uncertainty in the daily links data. So I just would like to note that we should be aware of these issues. Thank you.
Thank you. That's good to know. And is that on that difference seen in small tunas as well, or just the larger?
Small tunas, mainly.
Okay.
We've been keeping the juvenile like 1 year to 2 years old, because the experiment has been only 3 years for now. So we will be collecting data for large fish in the future, but for now we only have data for 1 to 2 years old fish.
Okay, well, thank you for that information. I didn't know about that.
Okay, any other questions? I have a question, Lisa. So often we don't have information on large fish. And so we're kind of extrapolating outside the range of the data for L infinity, which is something you said we shouldn't do. So what do we do if we don't know what L infinity is? How should we model that? What's the best way to eliminate any biases or represent the uncertainty, you know, that sort of thing?
Yeah, so it's a hard topic. I think really the biggest problem is with that von Bertalanffy. I think I always see it overshoot L infinity if most of your samples are smaller fish. So I think, you know, the model, you know, the growth cessation model you came up with, that's, to me, that's a very good option to anchor it down. I haven't seen a case where there's zero information on L infinity. I think often there's little information, but there's always a few old fish, and we should just, you know, be more on the cautious side and say, okay, well, the L infinity is probably the average around those few fish until you get more fish to say, actually, it's way beyond, because there's no reason why, unless you have a good reason to think that you're only catching the really, really small old fish. But we should just be within the bounds of realism with the few data points we have, in my opinion.
Okay, thanks. We should move on to the next presentation, which is Kai Lorenzen, who's going to give a comment on growth. And we're going to have a break and then discussion, so we can ask more questions on the for Lisa and other topics about growth after that. So Kai, can you share your screen, please?
Yes. Good morning. Can you
Morning, Kai. We can hear you clearly.
See me? Very well. Thank you for having me again. And thank you, Lisa, for a really excellent keynote. Again, I found myself agreeing with much of what you had to say. And what I want to do is just expand a little on specifically the topic of growth plasticity and variation in growth, which is sort of increasingly recognized as both a common phenomenon and one that can be quite consequential for fish population dynamics and fisheries dynamics. And it seems that in tuna, generally, people have been assuming that growth is not very plastic. But my question is that really true? Is it minimal, or is it hiding in plain sight? And I want to do that by just talking very briefly about, you know, recent developments and insights in our understanding of growth plasticity in fish in general and its implications. And I want to look a little bit at some tuna information and then get to better practices for growth modeling that account for plasticity. And really, this is a follow-on from the growth modeling comment that a whole team of us prepared for the Rome CAPAM meeting that was specifically looking at this topic. And we have to wrap this paper up and get it out, but look out for it, and I'll be drawing on what we did there. And basically, we looked at you know, how growth is influenced by environmental drivers, density dependence, and so on, and how that then filters through both the ecology and the dynamics of the stock and through the assessment approaches, and derived some recommendations for growth modeling in that light. And I just want to highlight some of the key points about the magnitude of growth plasticity. So we're finding that it's quite common that we find relatively strong plasticity in growth in fish populations. We find significant density dependence in growth in what just over half of fish stocks, compared to density dependence in recruitment in about 2/3 or a bit above that. there seem to be long-term trends related to global change in the order of sort of 1% in body length per year. And then, of course, there are evolutionary effects, or there may be evolutionary effects of fishing, but we actually expect those to be somewhat smaller. And plasticity has consequences. So growth, for example, contributed more to population variation in a good third of stocks that Christine Stavitz analyzed. Density dependence in growth can be more important source of compensation than stock and recruitment in some stocks. And in fact, about 30% of stocks that I looked at a while ago, that turned out to be a stronger a source of population regulation and compensatory reserve. And there's been a lot of interest in this area, particularly in the ICES world in the last few years, and people have been looking a lot at how density dependence in growth affects management reference points, find that it can make a difference of up to 40 or 50% in FMSY estimates. And that is positive differences. And so this has become a little bit of a politicized subject for the obvious reason that whether or not you find density dependence in growth and incorporate it, it can make a very substantial difference to your management reference points. And then finally, there are long-term trends associated with profound changes in ecosystem productivity and in ecosystem interactions. So in general, this is an important topic. The question is, so what about tunas? And my first question is tuna growth plastic? And I would say almost certainly yes. And that is based on two observations. One is that in the few studies that we have that have really looked at temporal growth variation that is likely attributable to plasticity, we find that the level of variation is commensurate what we observed in other fishes. And the other thing that I think is quite interesting is we are increasingly seeing tunas in aquaculture and in captivity. And in particular, we're seeing tunas being brought into capture-based aquaculture. And that allows us to basically look at how the growth of these organisms change when we put them in a very different environment. And it seems they're doing very much what I would expect a fish to do when we transfer them from a natural to a cultured environment. They grow faster to a smaller asymptotic size. And the reason for that is that they're changing the way they allocate resources. But it very clearly shows the level of plasticity that is innate in these tunas. How can it manifest itself? And there are several different ways: complex ontogenetic patterns, spatial variation, temporal variation, and long-term changes. And obviously yesterday and again today, we talked about those ontogenetic changes. And I was maybe a little overexcited about some of the magnitude, and I understand that may not be universal, but as also Elisa pointed out, we do see patterns like this in multiple stocks and multiple oceans, and it's definitely worth taking a closer look at those. There's actually a study by Emmanuel Dörtel on looking more into the underlying processes here, and this is a modeling study that was informed both by results from that captive yellowfin population in that IATTC maintains, and from wild fish, which shows that really this most likely, if it exists, is a sign of growth plasticity. And as I mentioned yesterday, if we do find patterns like that, we are likely to find a relationship between those growth transitions and some level of changes in mortality. We've heard a lot about spatial growth variation today, and some of that may be plastic, some of that may be genetically based, and I'm sure we'll talk more about that, but that's definitely a thing. Longer term changes, just a couple of weeks ago, there's a major study looking at Atlantic bluefin tuna growth literally over centuries that seems to have identified quite substantial medium-term growth changes, well, depending on what you call medium-term, long-term. And there was a modeling study out just this week or last week where basically using a bioenergetics model and looking at climate change impacts, and their prediction is a 15% decline on average in body weight of tunas by 2050. And so this is obviously a modeling study, but it shows us that we really should be aware of and looking into those changes in growth, and and see whether actually what this modeling study indicates will play out or not. So last thing, better practices. Well, several. One is, I think it's really important to recognize growth plasticity as an important biological process with very fundamental implications for fish population dynamics, fisheries assessment and management. And we should move on from the paradigm that growth is constant unless we find something else. Rather, we should embrace the idea that it typically will be plastic, but it could still mean that that is not really expressed. So it could be constant, but we should be aware that there's always that innate potential for change that plasticity brings. Continue and expand monitoring, pay attention particularly to ontogenetic changes in tunas, I think, and expand the use of hard part biochronologies to look at historical growth changes and perhaps understand drivers better. Thoroughly explore growth data for variation and changes in time and space. And Lisa touched upon that. I think it's often easier, at least initially, to do that outside of the stock assessment or even an integrated sort of growth model, because very often the flexibility that you have in those models is limited. And so it might be easier to do that outside to start with. Propagate uncertainty, clearly. We might want to run sensitivities, particularly for density-dependent growth, even if data are not informative. So we may not want to make the default assumption that it's just constant, but just as we might have sensitivities for steepness, also we might want to include sensitivities for density dependence and growth within the sort of range that is often observed. Use ensemble approaches to better represent multiple growth process hypotheses. And finally, make use of information from aquaculture and experimental work. And I think Carolina brought that up earlier, talking about the reproductive parameters. The same is true here for growth. And in fact, I think we can learn quite a lot from looking at captive populations, bearing in mind that their biology will not be exactly the same as in wild populations, but increasingly we get a good understanding of how those differ. And I think we can just learn a lot from that, and we're not doing it enough. And that's it. Thank you.
Yeah, thanks, Kai. Any questions for Kai? No questions? So can you explain again why you think the growth changes for sort of intermediate ages in, I don't know if it's the Atlantic or the Indian Ocean, but may not change in other oceans? Why is there oceanic differences in that change? So like in the Pacific, we don't see it, but at the Atlantic and in the Indian, we do, or at least in the Eastern Pacific, yeah.
So I don't know, I would like to ask you that question. It seems that there is these are associated with changes in basically the physiological capacity of the tunas to use particularly a deeper sort of colder water and the food resources in those. That was my understanding. But again, I'm not at all a tuna biologist, but it does seem that those changes coincide with points in their biology where they, their capacities change and their behavioral changes that go along with that allow them to exploit resources differently. But I would love to hear from people who know more about tunas.
Does anyone want to further comment on that? Yes, Simon.
I guess the example from the Indian Ocean, which was the one that you really picked up on, which Paige explained, was kind of explained away to some extent by being due to differences in two different growth cohorts, one growing faster than the other. So it's maybe that example is more an example of growth plasticity than of change in growth. I guess the problem is that we don't have enough sampling across enough cohorts and enough space to work out what's really going on across the whole population, and we're trying to represent the whole population rather than just particular parts of it. And it's also possible that the larger fish that are being included in those growth models are sampled from different areas than the smaller fish that we've got in those models. So we need to have enough data to tease out what's really going on.
Yeah, and I think, again, I sort of agree with Lisa's take that it seems we're not really sure. So, different analyses show different levels of those apparent transitions and different data sources. But at the same time, it does seem to be something that is somewhat more prevalent in the tunas than in some other species that we look at. And so I wouldn't completely discount it, but it does seem to, I think it just needs more attention because it may have important consequences. And I see Lisa's hand is up. So.
Okay, thanks, Kai. I think we'll break for coffee and still come back at 10:15 for a discussion on this topic so we can bring it up again. But I think there might be some behavioral changes in yellowfin, well, in tunas, like moving away from fads and also spatial movement that might change growth as they age. So I'm not sure if we should expect them to follow a standard curve through their whole life. Maybe we should be expecting some unusual curves. Okay, so thanks, and we'll come back at 10:15.